Hello, today I would like to speak to you about this question. With current gene mapping techniques, can I predict the location of a gene based on its composition? Uh, Hi, my name is Beth Fawcett, and today I'd like to walk you through a little bit of a genetics analysis that I have performed. First, let's talk about the background. So there are a couple of terms that you're going to need to know to understand this presentation. The first one is DNA. That's our genetic makeup. The next is chromosomes. Chromosomes are DNA. It's just that they're organized. The way that I like to think about chromosomes is that they are like a necklace. So the chromosomes are different strands on a necklace. And then the genes, which contain genetic information, but it's more specific, they actually code for proteins. Those genes are kind of like beads on a necklace. So we have multiple strands, we have multiple beads on each strand, and the strand can either the bead that is on the strand can either code for a protein or not. And this is called strand throughout this presentation. Now, proteins, each bead on those strands on those necklaces, are made up of amino acids. They're like the atoms in a necklace. So, those are the terms. On the bottom right hand side, that is a picture of a chromosome where the different white and gray areas are the different, what's called a locus. Now, a locus, the plural form is loci, are just areas. They're just locations where specific types of genetic information are stored. Now, the reason why I wanted to study this particular project and the way that I studied it is because it could assist with drug development, it could assist with individualized medicine, basically medicine that's tailored to your own DNA. It could lead to an advancement in understanding the mechanism behind chromosomal based disease processes, so diseases that affect your chromosomes, and a multitude of health applications. Now, for this particular study, I used a supervised learning technique, which means I had labeled data. Now, I knew the locus that was being classified, and I chose to study loci that only had more than 50 occurrences, and I specifically picked out chromosome 19 because it has 48 instances of loci with more than 50 occurrences, which meant I had a really good wealth of that labeled data to use. I did two different studies. I did a locus study and a strand study. In this case, locus being the location of the protein, and strand being whether or not the sequence that we're looking at is a protein or not. And I went through the stereotypical data science approach. We obtained the data, then we scrubbed it, then we explored the data a little bit, did some feature generation, then we generated our model, we optimized the model, and then I have some interpretation for you. The first thing that I want to talk about is this graph here. Now, in this graph, there, now you can see it a little bit better. We're looking at the molecular weight of our amino acids, which are the atoms and the beads on the strands on the necklaces, and their length. And a commonly known fact is that molecular weight and length are very related. And I used this knowledge to kind of power my study. The next thing that I want to talk about, I guess I should leave my face over here, is some things that I found. I found two things that were particularly interesting whenever I was looking at weight and length. Now, tryptophan is a really heavy amino acid. It's really heavy, and it was found pretty frequently, which I was surprised by. Methionine was also found frequently, but it's kind of like the start indication of this is a new protein. The next thing that I found was that tryptophan and cysteine, their weights were really high for how often they were found. What that means is that there was an increase of presence of these two particular amino acids in the proteins that we studied. And these, both of these findings, along with length and weight being related, really drove home my decision to start classifying using these length and weight metrics. And what I ended up finding was that in the case of the strand study, is this a protein or not a protein? I got a testing accuracy of around 72%, which means that there is something going on, but we can't really predict it accurately. We would need to maybe incorporate some other data to really drive at what's going on. I also found in the case of studying the locus, or predicting where a gene is located based on amino acid content, the length and the weight of the amino acids present, we got a testing accuracy of 99%, which means we could predict 99% of the time where a protein comes from. And this leads me to some future work that I'd like to do. I would like to continue to work on optimizing that strand study using some other different types of classifiers, maybe incorporate some additional chromosomes. I'd also like to try expanding my study. Originally, I mentioned some pretty strict conditions about chromosome 19 and more than 50 occurrences of a loci. I want to expand that to maybe everything present in chromosome 19. I'd also like to build a neural network, and I would like to build a chromosome, a full chromosome, from predicted values and test it against what we currently have available. I'd like to thank you for watching my presentation today. If you have any questions or any more, you'd like to look into things a little bit more deeply, you can visit my website or send me an email. Thanks for your time. Bye.